For this episode of Shareable Science Beyond the Blog, let's take a dive into why two individuals may have such a different response when infected by the SARS-CoV-2 virus. Welcome to Shareable Science. Science you can share. Two individuals can be infected with the SARS-CoV-2 virus and have incredibly different responses, ranging from no symptoms whatsoever to in the hospital on a ventilator. What can account for that wide range? Well, there are two broad areas, something to do with the virus itself and something to do with our response to the virus. Let's start with the virus itself. A straightforward possibility might be that individuals have different levels of virus early on in infection. Someone may be exposed to a high amount of virus, or someone may be exposed to a smaller amount. And there's some preliminary data that says your initial exposure amount may influence what your later symptoms are. Another possibility is that there are specific changes in the RNA genome, the genetic sequence of the virus, and that these changes may influence the virus's infectability, its ability to infect someone, or the severity of the symptoms. There are a group of scientists that are tracking these changes as the pandemic moves across the globe. And while they do see the virus mutate, it mutates relatively slowly, and so far there's no solid evidence that suggests that there are specific changes in the genome that correlate with severity. So that leaves us, you and me, the host for the virus. We already know that there are a specific set of risk factors that increase our likelihood of having a severe outcome. These are things like a pre-existing condition, lung disease, or obesity, or a compromised immune system, or being at an older age when you are infected. And we're still trying to understand exactly why those risk factors are prevalent, what their relationship is with severity of disease. But there's another growing area of research that actually suggests we might find some of the variation in response in our own DNA, in our host genome. So this could be differences that actually lead to how easy it is for our cells to allow the virus in, or the way that our immune response over or under responds to the virus. So this is a growing area of data. It's called the host genome impact. So how do we study the potential changes in our own genome? There are two broad ways that this is being done in the middle of the COVID-19 pandemic. The first starts with existing DNA biobanks. So there are groups all around the world that have recruited individuals to be part of the biobank. They've had a sample of their DNA. They know a lot of information about the individual's health. And scientists can go back to the people in the biobank and ask if send them a questionnaire, contact them and say, have you been infected? Have you knowingly been infected with COVID-19? And what was your outcome? And then they can take that information and they can try to track it back to genetic changes. The other approach that people can take is that they can start by recruiting patients with a COVID-19 diagnosis and ask if they'd be willing to participate in the study, usually by going to hospitals and clinics, and then obtaining a DNA sample from them and doing the work from that point forward. Researchers, my colleagues here at HudsonAlpha, are parts of these host genome studies and are looking to identify specific changes in our DNA that may give us a better sense of how well people fare, what the outcomes are. There are some early findings from this work around the globe, just a couple so far. One is in a set of genes called the HLA genes, the human leukocyte antigen genes. And these genes essentially help train our immune system to attack invaders and not attack our own cells. And there's some early data that DNA changes in these genes may increase susceptibility for infection by the coronavirus, or other variants may protect you from infection. And then there's also some data on the proteins that sit on the outside of our cells that the virus attaches to in order to gain entry. And changes in the genes that code for these proteins may also make it easier or harder for the virus to obtain entry. So the goal is by gathering this information, we may be able to identify individuals who are at a high risk of having a poor response if infected and individuals who are at a lower risk. And we could learn that information well before individuals are infected and give people a better sense of what their overall risk might be. I hope you find this information useful. This is data that we'll continue to add to over the next several weeks and months. Please share this with those around you. Thanks for watching. I look forward to seeing you next time. Stay safe.